Hello everyone, welcome to Global Compliance Panel's live webinar on GMP expectations for Phase 1 products. My name is Johnson and I'm going to be a host for today's session. On behalf of the Global Compliance Panel team, I'd like to thank you for being a part of today's event. Today's webinar will be presented by Dr. Stephen S. Kuhara. Dr. Kuhara, PhD, is the founder and principal of GXP Biotechnology LLC, a consulting firm that works in the areas covered by the GLP and GMP of drugs, biologics, and nutraceuticals. Dr. Kuhara has over 30 years of experience in supervising quality control laboratories, including an animal testing facility, and in performing GLP and GMP audits of internal and external testing laboratories. Dr. Kuhara has participated in development of drugs and biologicals through all phases of clinical research and final product production. We are honored to have such a distinguished person, such as Dr. Kuhara, with us to present today's webinar. Before we begin, I'd like to inform you of the program outline for today's training session. This webinar is for 90 minutes duration. First, Dr. Kuhara will take you through today's webinar, highlighting the areas that will be covered, and he would then share with you his presentation. Please be informed that all our participants, once part of the teleconference, have been placed on mute and will remain so until the Q&A begins towards the end of the webinar. We also request you to hold your questions back until the Q&A window begins. 10 minutes of time is allotted for the Q&A. If for any reason you get logged out of this training session or teleconference, please follow the same procedure to join in again. Now that we're ready to start, I request Dr. Kuhara to take it from here. Dr. Kuhara?
Okay, well, thank you, Johnson. I am going to talk about something that has been turning into a major pain for a lot of people. And this hits those of you who are working in development areas especially. There has been for a long time a belief, and I emphasize the word belief, in the industry that in early phase work you don't need to follow the GMPs. This is not true. It was a myth that was created by the fact that there are certain parts of the main GMPs that are not applicable to the work that you do in early phase studies. However, this does not mean that you can not use GMPs at all. And that's what we will talk about here. Now, early phase studies, as defined here, is, well, they're after the non-clinical GLP studies. And they're before the formal phase 1 study. Now, I will warn you, formal phase 1 study is when you actually get in and you get permission to go into a small number of usually normal human beings. Now, the problem here is that before you get to this stage, there are many, I guess they're sort of referred to as mini studies, where you do investigations quite often with so-called microdoses and other small amounts of material. And they're considered to be fairly safe in that the dose you give is not likely to be toxic and all this. But at the same time, if you go into humans, and this is the important thing to remember, the law, the Food, Drug, and Cosmetic Act says that if you go into humans, you must have produced the material through the use of the GMPs. And this is the whole catch here. Now, we're looking at the area between GLP studies and the formal, well, including the formal phase 1 studies. But after you conclude the phase 1 and you go into a phase 2 study, you have to go into the main GMP. Now, these studies that I'm talking about usually involve things like PK studies in normal people and ADME studies, again, in normal. And some of this thing has turned into sort of a mishmash. Um, here's the catch. I mean, you will get laboratories that test material from clinical studies and tell you that, well, they're using GLP principles. Now, GLP itself has some problems because there's clinical GLPs or, well, pharmaceutical GLPs, and then there's environmental GLPs and a whole bunch of odd GLPs that are out there. And some of the testing laboratories claim to be GLP compliant, but they're not GMP compliant. From my point of view, the whole thing's sort of ridiculous because people who are afraid to do GMP studies will then do so-called GLP studies when, in fact, the effort needed to do both properly is about the same. So you really don't save much by going to GLP studies at this level. But, that confusion is out there. So now, let's take a look. First of all, Q7A here are the GMPs for APIs, your active pharmaceutical ingredients, in other words, the bulk drug. And this has been around since August 2001, so it's quite well established. And there is a section in Q7A, section 19, which covers APIs for use in clinical trials. So the minute you go over into clinical trials, even the raw material that you use, in other words, the bulk drug, has to come under GMPs. And these are the GMPs as given in Q7A. Now, people will tell you that Q7A in the United States is nothing more than a guidance document, which is true, but FDA has said that everything that's in Q7A can be found in the main GMPs in 211, 210, 211. And the catch here, 21 CFR 210, 211, are the main GMPs, and you can look at the API GMPs and you can sort of see a correspondence. Now, it turns out that in early clinical trials especially, the material that you manufacture, your manufacturing procedures, usually do not need to meet full GMPs for various reasons. And so I'm showing you here a table, it's a table of contents actually, of what's in section 19 of Q7A. And we'll cover these sections pretty much quickly. But notice this, it states that not all of the controls are appropriate for manufacturing API. Remember this, APIs under the GMP, APIs are considered to be part of the GMP, have to be made by GMP. And the specific guidance here is what we will talk about shortly. But notice this statement down here. Once drug development reaches a stage where the API is used for drug product intended for clinical trials, you, the manufacturer, have to make sure that the APIs you use are in now suitable facilities using appropriate production and control procedures. That statement there covers a whole area of work, actually. So let's go ahead and talk about this. Now, the first thing is that you have to have a quality unit. And again, the quality unit should be independent from production. And now some of the testing functions commonly performed by quality units can be done in other organizational units. Now, later on, you'll see that allowance is made at the API level for the idea of having some of your manufacturing people do QC testing, for instance. But there's, a bunch of conditions attached to it. Now, what you're going to find is that Q7A came out way before the GMPs for phase 1 products. And what has happened here is that the GMPs that we'll talk about later have been set so that they sort of match up to the GMPs in Q7A. Now, again, quality measure system for testing of raw materials. Now, one of the things, let's take a look at this earlier thing here. They talk about control of raw material. This is not the raw material that goes into your final pharmaceutical. These are the raw materials that are used in producing the APIs. So this is, one of the important distinctions here. So we not only have to check the raw material that goes into the finished pharmaceutical, but also the raw material that goes into the manufacture of the API. Now, this can be a problem for some of you because you are contracting with other organizations to make APIs. And this means that you must make sure that that organization has control over the raw materials that it uses. And this can be a problem because in some countries and some types of products, especially biologics, establishment of the quality of the raw material can be a real problem. Now, you have to evaluate all of your process and quality problems, and the labeling, again, for use in API should be appropriately controlled. Now, I will suggest to you that, your controls here are GMP controls. And there's no point in saying that, well, this is GMP for API, so we don't need to do this. And then, oh, this is GMP for finished products, so we need to do these additional things. Actually, why don't you just go to one system? It saves you a lot of stress and bother in the long run. Your equipment and facilities have to be controlled, and including things like small-scale facilities. In the early stages of your work, and especially when you produce material for phase 1 clinical trials, you may be making small batches, and these batches may be made in a laboratory setting rather than a real manufacturing setting. But even then, in your small-scale facilities, you should have procedures in place to make sure that you've got equipment that's calibrated, clean, and suitable for intended use, otherwise known as qualified. Now notice this, when it says procedures, they're talking about SOPs, which means somebody better get down there and start writing these SOPs for your equipment and facilities. Again, in your facilities, you have to make sure that your material is handled in a way to minimize risk of contamination and cross-contamination. For people who do preps at the lab scale, one of the problems here is that a lot of times in your laboratory, you're going to have people that are doing other kinds of work. And there is always a possibility that their work or the material that they use could contaminate the material that you're preparing for the clinical trial. So you have to make sure, and especially if you go out and evaluate your API manufacturer, you have to make sure that the handling and procedures minimize the chance of cross-contamination from other work. Your raw materials here at the API level should be evaluated now by testing, which is the usual thing, or you can receive it with a supplier's analysis, otherwise known as a certificate of analysis. But remember, C of A's by themselves are never enough. You must have additional identity testing. And, the identity tests are fairly simple and straightforward, but that has to be done. You can't just accept on a C of A alone. And this is true for finished product raw material as well as for API raw material. Now, if the raw material is considered hazardous, then you can go on the C of A alone, the supplier's analysis. But remember that when you accept on a C of A, the testing that is shown in the C of A must satisfy the requirements for your product. So if the C of A, for instance, if there's a one of the better examples here is that you have a requirement for low levels of heavy metals, or specifically, say, for low levels of lead or mercury or something like that. And the certificate of analysis doesn't show that, does not show analysis for heavy metals or something like this. Then the certificate of analysis alone is not enough. You must do additional testing. And you may have to send the thing out for heavy metals testing if necessary. And on top of that, you still need to do the identity testing. Now, some cases you can determine suitability, and this is also true for finished product raw material. You can do it by use testing. In other words, you test the material in your process at small scale, and this is use testing. And if it's acceptable in that kind of work, then you can proceed with using it for your actual manufacturing. Production should be documented. Now, at this stage, you can have documentation in laboratory notebooks or batch records. And it says other appropriate means. This would include electronic records that you use. Documents include information on use of the production material, equipment, processing, and scientific observation. So this is pretty much like a regular batch record here. Expected yields are allowed to be more variable and less defined. And here's the thing, investigations into yield variations are not expected. In other words, if you have a OOS, or out of specification result and the specification would be a yield specification, you don't have to go in and investigate it if there's yield variation. And this is at the API level, remember, this is not at the product level. Process validation is normally inappropriate, remember this. This is at the API level because they're talking about small numbers of API batches. Now, here's the thing. If you go on to batches produced for commercial use, in other words, when by the time you've got a licensed product, then it refers you to Section 12. Now, we're in 19, this is 12. Section 12 of Q7A talks about process validation and what is expected for APIs. So you need to go to that when you reach the point where you're going to commercial production, but not necessarily when you're dealing with clinical trials. Changes are expected. You see, this is the thing. In your early clinical trials, you may discover things that will cause you to have to change some of your specifications, your procedures, and all of this, especially when you're going to scale up. And now remember, this is talking about all of your clinical trials, not just the phase 1. And so as you go from phase 1 through phase 3, especially your so-called pivotal trials, you're going to start to make larger and larger lots of material. And sometimes when you're doing that scale-up, that's when you discover that there are some problems with scale-up, and you may need to adjust your procedures and processes to allow for this. However, remember this, every change in production methods, specifications, or test procedures should be recorded. Your laboratory controls, now here's a catch here, and I want you to remember this statement. In Q7A, it says that analytical methods to evaluate API for clinical trials may not yet be validated. Nonetheless, they should be scientifically sound. Um, you know, it's one of these statements that's sort of self-contradictory because how do you determine that they're scientifically sound? Well, one of the ways you do it is by validation of the procedure. Now, the assumption is made here in this part of the regulations that at this stage you may be, well, producing APIs, and actually because you're in early stages, the API manufacturing may not yet be well developed, and your test methods may not yet be well developed. And so at this stage, you may not have fully validated analytical methods. But remember this, your analytical methods are part of your senses. As you know, they're your eyes and your ears. This is how you know whether or not you've got good material. And if your analytical methods are not quite up to snuff, this is like going around with bad eyesight, for instance, or bad hearing. I mean, you're going to tend to miss things. Now, a system for retaining reserve samples of all batches. And this is very important for anybody working with material at this stage. Keep retention samples. Now, I admit, for some of you, especially in early stages, the material you're using could be very expensive. And keeping retention samples is going to be expensive for you. But I will tell you that in the long run, you will be glad you kept these retention samples. Because without these samples, later on, it may be very difficult to prove that the material you used in your early phase studies had the same characteristics as the material you're going to use in your in your pivotal clinical trials, in your full-scale clinical trials. You should keep for an appropriate length of time after approval, termination, or discontinuation of an application. So we're talking about IND at this point. Expiration and retest dating applies to existing APIs used in clinical trials, and it does not. Now, 11.6 is the section of Q7A that deals with stability testing and expiration dating. And it says for new APIs, however, you may not have the information, and so 11.6 does not apply in early stages of clinical trials. Okay, so this would be where you're first starting to make the API. You may not have these retest and expiration dates at that point. But when the APIs become sort of established, you should have expiration or retest dating on the material. Documentation. Remember, document everything. The whole business sort of floats on a sea of documents. And you should have a system in place to make sure that your information is documented and it's available. Development and implementation of the analytical methods should be documented. System for retaining production and control records should be used. In other words, you better have archives or at least a documentation system that retains all of this information. There's a couple of catches here because in this early stage, when you're first starting to develop your product, you're going to find that a lot of the quality by design efforts in what is known as Q8, which is the development guidance document from ICH. A lot of these provisions apply, and the impact, however, may not hit you until you get into later stages or you actually file an IND, for instance, or I mean rather an NDA. You file, or a BLA where you're filing for a license. The problem here is that this documentation may be for very early work, that could be many years before you file the big application. And yet, if you screw it up at this stage, later on, when people are preparing the license application and everything, if they can't find the documentation that's needed, there can be many problems because you've got years to go back and check. So, I really tell you that at this stage, make sure you've got a good documentation system. Okay, that ends Q7A. Now, that talked about the APIs that you're going to use to make the product you use in clinical trials. Now, FDA came out with I guess it's an amendment to the GMP, basically, that says here, and this was published as a direct final rule. It came out in the Federal Register in 2007. And basically, the summary was this. They concluded that the full CGMP regulations should be exempt in most phase 1 investigational drugs. Now, remember this. They're talking about phase 1 investigational drugs, nothing else. FDA continues to exercise oversight. In other words, they're going to regulate you by other means, mainly through the rules for the IND. And remember, this is 21 CFR 312 that covers the IND process and the early clinical trial material. The rule became effective in 2008 on September 15th, and well, they say several reasons. We're talking about the rule here, and here's the catch. They tell you, well, they're trying to justify this whole thing and saying that they still have statutory control over the drug, even though it's being used in a clinical trial, and they've exempted you from GMP. Now, it's kind of strange. It does not. It must be in conformity with current good manufacturing practices because of these statements in the Food, Drug, and Cosmetic Act. Remember, USC, United States Code, is the federal law. This is codification of federal law. And the 21 USC covers the drugs, the Food and Drug Act. Now, second, they have authority through the IND process, and they talk, now this is 21 CFR 312 is what they're talking about here, not USC. This is the Code of Federal Regulations. And it tells you that, well, okay, you still have to submit CMC information, and this tells FDA of what you're doing to ensure safety and quality of the drug. And remember this, you are subject to auditing or inspection by FDA, beginning at the IND level. They can step in and say, okay, are you doing what you said you're going to do in the IND? And finally, here's the actual modification of the regulations. The modification was made in 210. Now remember this, GMPs are actually in 21 CFR 210 as well as 211. Now 211 is considered to be the main part of the GMP, but 210 is the introduction. Excuse me. Oh, my throat is being attacked by something. Okay, now, this is 210.2C. And the modification was made here. It said it's subject, they're described in 31221, the investigational drug. They're still subject to the Food, Drug, and Cosmetic Act here. However, production of the drug is exempt from compliance with regulations in Part 211. 211 being the actual, manufacturing regulation. Now, no. However, and always watch out for however, it's an FDA statement. It does not apply to an IND once the drug has been made available for use or by the sponsor in a phase 2 or phase 3 study. So as soon as you cross over into phase 2 and phase 3 study, the drug becomes subject to the full GMP. And these exemptions only apply to a drug that has never been used in a phase 2 or phase 3 study or ever given a license. So remember that this is for early development work on a new drug that has not been taken through development by another laboratory. For instance, if you take say a licensed drug that's out there being manufactured by a big pharma company, for instance, and you're going to make that drug as a generic with the idea of using it for another indication that's not, indicated by the major manufacturer. You will want to step back and do a phase 1 study just to establish that it's safe at the dosage you're going to use and with the formulation you're going to use. At that point, however, because the thing was licensed, you still have to do full-scale GMPs in producing the material for this phase 1 study. If, on the other hand, the drug is completely new, it's never been through phase 2, 3, and onward studies, then you're exempt from the 211s. That's the whole point of this thing. But once it's been made available or it has been lawfully marketed, in other words, you've got a license for this thing. Once that's happened, you must comply with 211. Now, not only happen by you, but happen in the industry. Okay, so you can't claim that, well, for us, this is a new product and we're taking it into phase 1 for the first time for this indication. If the thing's been out there on the market, it's still subject to it is now subject to full GMP. Now, here's the companion guidance that went with that previous rule. Okay, here's the thing. Here's the rule. It's the amendment to the rule, to the GMPs. Now, this guidance here was added to tell you what are the GMPs that you have to follow. Now, remember that. You were exempted from 211, but the Food, Drug, and Cosmetic Act still says that you must follow GMP. So what FDA did was that it came back with this guidance document that says these are the GMPs for phase 1 products. And this was dated November of 2007, but the real effective date on it was July 15th of 2008. And notice that it comes in from the drugs people, CEDAR, comes in through CBER, and it's the Office of Regulatory Affairs. Notice there's no drugs people, no CDRH, and there is no CVM either, veterinary medicine. Now, here's the introduction to the thing. Guidance applies as part of CGMP quality control principles to the manufacturing of phase 1 drug. And it goes on to, basically, they're just telling you that, okay, even though you're at phase 1, you still need to comply with good quality practices and the like. Now, this one is important. It does not apply, this guidance document, this new GMP, do not apply to first human cell or tissue products that are solely under 361 of the Public Health Service Act. For those of you who do cell or gene therapy, these products are known as 361 products, and 361 products do not have to meet the requirements of the GTPs, the good tissue practices procedures that are found in 21 CFR 1271. And they are considered to be a special class of minimally manipulated cells. Minimum manipulation means that, well, for instance, blood, you take it out, and you might even separate the plasma from the packed cells and then dilute it with saline or some other physiological solution and then give the cells back to the patient. Okay, that's considered minimal manipulation. Now, if you take those cells and treat them in some way, including going and doing gene therapy and modifying the cells, then they come under the full 21 CFR 1271, and they are not exempt. These are the minimal manipulated cells. Clinical trials for products for device approval, in other words, devices, things that are covered by the 820, 21 CFR 820 GMP. IND products for phase 2 and phase 3, already approved products that are now being used in phase 1. And, remember this, if you change the indication for a drug, you need to go back and do new clinical trials for the new indication. And it may be necessary to do a phase 1 here. And then PET drugs, positron emission tomography drugs. There's a curious thing here, is that it refers to GMPs that do not exist yet. 21 CFR 212 will contain the GMPs for PET drugs, but this is yet to be published. General guidance, again, appropriate QC procedures in place. Adherence will be through, guess what, well-defined written procedures. In other words, a whole bunch of SOPs. Adequately controlled equipment and manufacturing environment. In other words, you will have procedures for controlling the manufacturing environment and the equipment used in the manufacturing. In other words, cleaning, maintenance, all that sort of stuff. And then accurately and consistently recorded data from manufacturing, including testing. So this has to be, you have to have a mechanism for this. Your manufacturing environment, you have to consider the hazards associated in the environment that could adversely affect the quality. Now, the main thing that they're worried about here is where you manufacture in laboratory facilities that are not solely designed for the manufacture of the drug. Okay, because you have other things going on in a laboratory, and there's good chance of cross-contamination or contamination with other substances that are present in that working environment. And you have to show that you're controlling this. They recommend that the following steps is to establish manufacturing environment. Now notice this, when they say we recommend, if you don't follow the recommendation, you should be prepared to justify why you have not followed the recommendation. And this justification had better be good. You can't just say, well, we didn't feel like it, or the equivalent in terms of 500 words or less. The thing is, if you don't follow these procedures, remember, you have to have a good justification. You have to have a evaluation of the manufacturing setting. In other words, you better go in there and think about all this stuff and write up a evaluation. Appropriate actions prior to and during manufacturing to mitigate or eliminate potential hazards. This is the thing, under quality by design, when you get to this early stage of manufacturing, when you're doing development, you should have done a risk analysis. And here is, your hazards and all this. What did you do about the hazards that you identified? Now, at this point, you might say, well, I don't know what the hazards are. And they then go on to tell you what they would like to see you do. First, adequate work areas, properly equipped and controlled for these specific operations. Given the diversity of not all environments may be acceptable for the manufacturing of specific phase one. In this case, you should use more suitable facilities. Now, one of the things that has arisen, especially nowadays where we have drugs that are getting to be more and more potent, there are some drugs known that when you dilute them down into the actual form that you administer them, they're perfectly safe. Botox is a good example. However, in the concentrated form, where you're producing it at the API level or producing it initially before you dilute it down, these drugs can be very toxic. And the guys that are working with the drug may be well aware of that. However, if you contaminate the rest of the lab, the other lab workers may not be aware of the dangers and could easily get sick or die or all that sort of stuff. In that case, these drugs, it may not be suitable for you to be manufacturing these drugs in a laboratory that's kind of open with access by a lot of different people. And you may need a controlled environment in that situation. Contractors, basically under CGMP, if you use a contractor, then the contractor is held, well, both you, the sponsor of manufacturer and contractor, are both responsible for assurance that GMPs are followed. In this case, the phase one drug. Assurance is achieved by the following. And again, we recommend that you assess the contractor. Don't just pick somebody out of a directory or one of these things of, well, Joe worked with them at another company, so let's try them. You have to assess them to make sure that they have proper quality control function. Now, recommended CGMP must be in place. Manufacturers should establish controls based on identified hazards for the manufacturing setting that follow good practices. Now, the following controls are applicable to manufacture of phase one IND drugs and in specific manufacturing situations. Remember that. Personnel, again, pretty much what you'd expect. Personnel have to have education, experience, and training, or any combination thereof. Now, remember this, combination. It doesn't mean that you can eliminate one of these in favor of something else. For instance, you cannot say that a person has education and experience, so they don't need to be trained in your specific procedures. This does not happen. And so all three elements have to be present to perform the assigned function. And in particular, you should have experience, appropriate experience to prepare the phase 1 drug, be familiar with QC principles and acceptable methods. There's a lot of people who come straight out of university labs who've got the experience to make the drug, but are not familiar with quality control principles. And and this is the problem, is that in a lot of companies, this is what you need to do, is to train these people in the quality control principles and the GMP requirements. Now, notice this, as described in this guidance. Okay, so you're limited to the GMP for phase 1 products here. You have to have a written plan, describes roles and responsibilities for QC functions. Now, the written plan should have as a minimum the following things: responsibility for examining, responsibility for review and approval. This is pretty much what the quality function is supposed to be doing in a regular manufacturing setting. And this is, being brought back into the phase 1 GMP. Responsibility for releasing or rejecting each batch. Now remember this, manufacturing has sort of an inherent conflict of interest for releasing product. Of course, they will tell you they always make good product. So they will tend to be biased in favor of the acceptability of the product. And this is why you need this second pair of eyes to verify. Responsibility for investigating unexpected results or errors. This is the OOS problem, the out-of-specification test result. And remember, it's not just out-of-specification test results that need to be investigated. It's any unusual occurrence that must be evaluated for its impact on the product. Now, again, we recommend that you assign an individual to perform a QC function. Now, later on, they'll say that this is not necessarily true, but there's always the but. We'll talk about that. Now, here it is. In limited circumstances and depending upon the size and structure of the organization, if you have a small, say, 5-person operation going, you may not have an individual that you can designate as a purely QC person. So they will allow you at this stage, all QC functions may be performed by the same individuals who perform manufacturing. It may be necessary to have a person do both manufacturing and QC functions. Now, in such circumstances, they strongly recommend that another qualified individual that's not involved in a manufacturing operation conduct a periodic review. Usually this would be a supervisor, not necessarily your manufacturing supervisor. This could be a vice president, for instance, who steps in and at that stage in the development of your company is acting as a QC reviewer. And this is possible, but they do recommend that you don't just leave it in the hands of these people, that you still have an independent reviewer. And again, when activities such as testing are performed by manufacturing personnel, you should have adequate controls. Like segregation of testing from manufacturing, so that you don't have a possibility of cross-contamination or other event that can negatively affect the test result. Facilities, you should have the following described work area: sufficient space, clean environment. This is a problem for some of you who work in these very small startup companies, where essentially what you've done is you're renting what used to be a 7-Eleven store, and you're trying to run your whole manufacturing thing in that space. Basically, what FDA will tell you is that if you don't have enough space to do all the work you need to do, you need to get it. This would be like somebody arguing that your university cannot afford the land, therefore your football stadium is only 80 yards long. They're not going to let you play football on that thing, and neither will FDA allow you to do work in a area that's cramped and too small. You should have appropriate lighting, ventilation, and heating. And remember, these things, some of these things actually go out of the hands of FDA and cross into occupational health. And believe me, you're going to get checked by OSHA from time to time, especially if you have a major accident. You have to have cooling, plumbing, washing and sanitation facilities, equipment needed to maintain air cleanliness. Now remember this, even if your product doesn't have to be sterile, you should still have clean air. The classical one here is in some foreign countries, for instance, there's a steel mill in the vicinity of the manufacturing facility. And the ash or dust that's produced from steel manufacturing has a high level of mercury in it. And that ash or dust can get into your facility. So you need to have filtered air incoming. And, all of these things have to be controlled. You have to maintain your air cleanliness. I mean, really, you cannot have dust in your manufacturing area. And you should have, especially for things that need to be sterile, you may need to have laminar flow hoods and all of this. Again, you will have appropriate equipment. It has to have inert surfaces, cannot interact with the drug. Recommend you identify all equipment that's used. This is the same as in the regular GMPs, where you have identification of each part or each piece of equipment, including tubing, that's used in your manufacturing process. Excuse me. If you do sterile products, Section 6C of Q7A will cover that. And, for making investigational drugs that require aseptic processing. And this is basically any parenteral drug. Use of procedural controls in a facility promotes orderly manufacturing and all this. Procedural controls, otherwise known as operating SOPs. Control of components, containers, and closures. Again, you have to have written procedures, otherwise known as SOPs, on how you handle your raw material, your components, raw material being components, containers, and closures that are used in the manufacturing. And, material should be controlled until you have examined or tested. This is the, raw material, incoming material testing, and released for use in manufacturing. Remember, the people that do this work are supposed to be the quality people, the QC people. And you must store in such manner as prevents degradation or contamination. Now, you should be able to identify and trace all material used in a manufacture of phase 1 drug from receipt to use in manufacture of each batch. Now, remember this. These tracing mechanisms must be both forward traceable and backward traceable or reverse traceable. In other words, if I go into your warehouse and I find a drum of sodium chloride with a certain manufacturer's lot number on it, I should be able to ask you, if I notice, for instance, that material has been reused from the drum, you have to be able to tell me where that material went. What was it used for? Account for the missing quantity. At the same time, if I find a product that's being, say, used in a clinical trial, and there's a question about the purity of that material, and I start tracing back, and I say, okay, where did the sodium chloride come from? Who is it? Where was it used? And who manufactured it? Who did you buy it from? From the lot number of the clinical material, I should then be able to trace it back to the manufacturer's lot number of the material you had in storage. So all of these things, you need your receipt. Here's your minimum information for your raw material. When did you receive it? Quantity that you received, supplier's name, material lot number, storage conditions, and the corresponding expiration date. Now, it may be that you may not have your own expiration date, in which case you may just adopt the expiration date that's given to you by the manufacturer. Also, remember this receipt date. Okay, a lot of times we have people that argue that, okay, on Friday we receive, say, 1 kilo of material from a certain manufacturer. On Monday, we receive another kilo of the same material, same lot number from the same manufacturer. The procedure well, a lot of lazy people, or, well, I shouldn't say lazy, maybe they're just sloppy, but a lot of people will combine the two and say that, well, this is the same material, so we don't need to handle it separately. The rule in pharmaceutical manufacturing is the fact that you have received them on 2 different days means that they have a life history. The 2 items have a life history that is different. They're not really the same material anymore. We don't know what happened to one of the bottles over the weekend when it was being held. So within the company, for manufacturing purposes, you need to treat these things as 2 different lot numbers, internal lot numbers of the same material. And so, you need to know all these things. And your storage conditions. A lot of times people have expiration dates that are based on stability studies of their final product. But the question is, how stable is your raw material? Your incoming component, how stable is it? You should have acceptance criteria for the attributes of each material. All relevant attributes or acceptance criteria may not be known at phase 1. And this is, quite true, because at phase 1, you're still sort of learning about your product. And it may turn out that there's an attribute that's important for your final product, but because you're still in phase 1 work, you may not have seen this problem, or the requirement may not have become obvious at this stage. So it's not unusual for you to add requirements as you go along. But it just basically recognizes the fact that you may not know all the relevant acceptance criteria. Now, all of this it says it'll be reviewed in the IND. Yes, FDA will review your criteria in the IND that you submit, say, for your phase 1 study. And again, it says examine the C of A to make sure it meets your established acceptance criteria for specific attributes. And, again, we can go back to freedom from, well, I remember a company that was purchasing a botanical as its raw material. And one of the problems here was that it was being purchased from a country that does not have, say, stringent controls over things like pesticides and herbicides. And every so often, they would get a lot of material that contained a high level of contaminating pesticides. And the catch here was, of course, that the company was smart enough that it was testing for the pesticide, and it would periodically reject them. But the certificate of analysis that it got from the supplier did not even mention pesticide testing. So it became a problem for them in that they had to go out and test for pesticides in these botanicals. Again, documentation should have information on sourcing and test results for adventitious agents. You know, sometimes, especially with biologics, you're going to get material in that's carrying adventitious agents. One of the big bugaboos these days are viruses. And especially with some biological material, including things like bovine plasma and the like, you can be carrying an undesirable virus. And you need documentation. And if that's incomplete for that attribute, you should test for it. And for each batch, you should perform confirmatory identity testing. See, the identity testing part of this is very important because you cannot believe, you know, you can't simply say, okay, the label says sodium chloride, therefore the contents must be sodium chloride. You have to actually perform an identity test that tells you that, yes, it is sodium chloride in that bottle. Manufacturing should follow written procedures and processes, otherwise known as a batch record. You know, your manufacturing process control procedures that has a record of the manufacturing details and everything, record of changes. This is the important thing, because you can have a perfectly good batch record, but over the course of time, if you have changes here and changes there, after a certain period of time, you're going to have the actual procedure and the batch record no longer being the same thing. And at which time you had better be able to account for the changes that took place and the rationale for the changes. Your record of microbiological controls that have been implemented. Now, they're talking here about production of sterile process phase 1 drugs. Now, remember this, even for drugs that are not required to be sterile, say like an oral dosage form, you still have to be able to show that there is no contamination by what is known as undesirable organisms. A list of undesirable organisms can be found in the USP and in the EP and the JP, depending upon which pharmacopoeia you're following. However, you know, you can't just say that, well, it doesn't have to be sterile, so we don't need to check for undesirable organisms. After all, nobody likes to get diarrhea from the product that they're taking. And, you know, you should follow recommendations, and especially if you're making a parenteral, for aseptic techniques and the control of material and all this. And that should be reflected in the records, by the way. Laboratory tests. Now, you remember the earlier thing on Q7A said that you may not have valid, may. It's not a permissive thing. It's a conditional thing. Maybe you don't have validated laboratory testing procedures at phase 1. However, notice this. If you go through the testing of material, in-process material, packaging, or drug product, should be scientifically sound, which is what the other Q7A said. You know, it doesn't have to be validated, but it should be scientifically sound. Now, in this GMP, it says, okay, scientifically sound, that is specific, sensitive, and accurate, suitable and reliable for the specified purpose. You know, if you put that all together, you have the definition of a test method validation, or what is a test method validation. So it becomes kind of strange here in that in one place they say that you don't need to validate it, but then they put up conditions which basically say that you should validate your test method. You have to have SOPs, you have to have written SOPs, you should maintain records of all test results, procedures, and changes in the procedure. And remember, changes in the procedures can be what may be to you fairly minor. But from FDA, from the regulatory standpoint, they're important changes. I had a problem once with an FDA inspector where I ran out of a potassium salt of a reagent and I substituted an ammonium salt of that same reagent, and everything worked perfectly fine. But then they said that on inspection, they said that, well, you changed the procedure, you used a new chemical, and there's no written justification for why you made the change or what its effect was. Did you check for the effect? And, did you evaluate? Now, you perform laboratory testing to evaluate quality attributes and those that define the following. For known safety-related concerns, you should have specs. And for phase 1 investigational drug attributes, all relevant, again, they say all relevant acceptance criteria may not be known. But again, the information will be reviewed in the IND submission. To ensure reliability, you should have calibrated lab instruments. Sometimes this is nothing more than, the usual thing of you come in the morning, first person to use the pH meter, checks its calibration and makes appropriate adjustments and everything, sort of a routine thing. That's fine. But if you have, especially more complicated instruments and everything, you have to make sure that the calibrations are done and the maintenance is done according to SOPs. Now, we recommend personnel verify equipment is in good working conditions when samples are analyzed. This is the rule that's used in a lot of QC labs, which is that before you use an instrument, like for instance a spectrophotometer or a GC or HPLC, before you start the procedure, the first thing you do is to check and make sure that the instrument calibration, it's not due for a new calibration, that it's within its calibration period, and it's within its maintenance period, that it doesn't need maintenance. If either of these are expired, then you need to do this before you use it in your testing. Retain a representative sample. Again, these are your retention samples. Now, recommend sample consists of quantity adequate to perform additional testing. And the example here, for instance, twice the quantity necessary to conduct release testing, excluding testing for pyrogenicity and sterility. That's because we know that these two attributes can change over time upon storage. Now, again, recommend that you appropriately store. In other words, you got to know something about the stability of these things, and retain the samples for at least 2 years following the termination or withdrawal of the clinical trial or IND. Stability, you should have, and you should initiate a stability study. Now, the thing here is this. Notice, all you need to do is to establish the stability of the investigational drug during the clinical trial. In other words, from the date of manufacture through the date of last administration. Now, for some of you, this can be a problem. Set up your study for longer than the clinical trial will last. And I'll tell you why. a lot of times people will tell you, well, we can run this phase 1 clinical and it's only going to take 6 months. And so what happens is that you manufacture your product, say in March, and then they don't launch the clinical trial until June. Now you're going to run a 6-month clinical trial that's going to end in December. And at that point, if you only have 6 months of stability data, your product will expire in the middle of the clinical trial. Okay, so what you're looking at here is your total time, date of manufacture to date of last administration, which is the other problem, because a lot of times they will tell you that, well, okay, we have a 6-month clinical trial, phase 1 especially, but, oh, gee, we weren't able to get sufficient numbers. So it's going to take us another 2 months to get enough people or get the right volunteers and all this sort of stuff. In which case, again, your product could outdate in the middle of the clinical trial. So make sure that this stability period that you go for, where you're actually doing the study, that this period is much longer than what they tell you the clinical trial is going to go through. Packaging and labeling, again, suitable packaging, and you should have procedures for controlling the packaging, labeling, and distribution. Now, at this stage in phase 1, you probably won't have a major packaging and labeling operation. I mean, you won't have that many vials, and they will even tell you that you may need to just go to manual filling and packaging, and at which case you need to do visual inspection of your labeling and packaging operation. And this would be your QC review and verification by a second person, especially where you have a potential for mix-ups, and where you, for instance, you can, and this has happened recently in some very major studies, where they have mixed up the placebo with the active drug. And, in a blinded trial, a lot of times you don't know if you're using the placebo or the active. And if the packaging of the product was such that they mixed it up to begin with, you're going to get some very strange clinical trial results. So, make sure that this whole thing, even at phase 1, that this is carefully controlled. Finally, packaging, labeling, and distributing. Now, remember this, distribution includes transport of the drug covered by the guidance into 2 clinical investigators. Excuse me. In one case that I had some experience with, what happened was that we made the drug and we put it on the plane, and the airplane flew high and lost control of its heating system. We had a liquid drug in vials, and they froze. And upon thawing, they cracked all the vials so that the clinical site received material that was completely, well, it was sort of a mess, because solution had leaked out of the vials and soaked the packaging and everything. And the point here was that we did not do a proper shipping validation. We did not realize that the plane that we had put things on did not have the proper heating and temperature control in its cargo compartment. And this is the thing. You are responsible for this. Remember this. When you make the product, your responsibility goes all the way to the point where it gets administered to the patient. So it's part of your job to make sure that the clinical trial site receives the product in good shape. So, make sure you you do your controls and testing all the way to the final product site. And companies are known, for instance, to take material at the clinic, at the point of delivery. They'll take an extra vial and send it back to the company for testing, just to verify that the material got there and went through all the handling and everything, and was still suitable for intended use when it was administered to the patient. Here's your record-keeping requirements. And again, this is not a thing that, I don't know, the problem here is that a lot of you guys, I guess in grad school or something, they tell you you don't have to keep records. And, those of you who got PhDs and went through a study and then where you had to go and work for 4 years and then try and look at your lab notebooks to write up your PhD thesis, you know that there can be a problem with record keeping. A lot of the stuff you did in the early stages of grad school no longer makes sense to you 4 years down the line. And this is the problem, is that you have to have this record keeping, especially at this level, because, let's face it, your phase 1 study could easily take place 5 years before you go to file your NDA or BLA. And in that period of time, you need to be able to check your records to make sure that nothing much has changed. So all of these things need to be in your record. Maintenance calibration, manufacturing records, analytical test methods that are results that are related to those things, distribution records, your QC functions, and that's in 5B. This is 5H. Component records, your raw material that went into your product, deviations, investigations, and any complaints that you got. And you must retain records for at least 2 years after the marketing application is approved. Now remember this, it's not after you submit it, it's after it's been approved. So if it takes a year to get this thing through FDA for approval, you've got 3 years essentially. And if it's not approved, again, 2 years after the shipment and delivery of the drug for investigational use has been discontinued and FDA notified. So there are record retention requirements here. Okay, well, thank you very much for your attention and everything else. Johnson will now take over and we will entertain questions.
Thank you so much, Dr. Kuwara, for the wonderful presentation. Also, would like to thank all our participants for cooperating with us. It's time now for the Q&A session to begin. In case you have any questions, please feel free to click on the palm-like icon, which is a small icon at the bottom of your participants panel, so that I can unmute your lines and you can ask your questions verbally. Or you can also go ahead and post your questions on the Q&A panel or my chat panel, and I can forward it to our presenter to read it out loud and have them answered. In the meanwhile, we request you to sincerely share your feedback with us. The feedback form will appear right now on your polling panel. It has about 8 questions, mostly multiple choice in nature, wouldn't take more than 2 minutes of your time to answer. The feedback form will remain open till the end of the session.
Yes, in addition, I would, well, Johnson, you already stated this, I think, earlier on. But, I've been to webinars and seminars, and I know it's a very common thing where at the time they say, are there any questions? And you're sort of sitting there not thinking of anything. And then later on, say an hour or even a day later, you start thinking, gee, I should have asked that question. Well, if that happens to you, feel free to send the question to GlobalCompliancePanel. And Johnson or one of his people will make sure that the question reaches me, and I will respond, fairly soon, if at all possible. And so, just because you don't ask a question right now doesn't mean that you're blocked from asking the question later.
Thank you, Dr. Kuwar. Well, Dr. Kuwar, we have one question that's come up, actually two.
Okay. Let's see. I'll open my chat panel here.
Mm-hmm.
Okay. I have a question. Is it necessary / recommended for a facility to register as a manufacturer with FDA if engaged only in the manufacture of phase 1 drug products which meet the exemptions specified? Yes and no. Let me put it this way. My position is that you should always, register with FDA. Now, for instance, there are certain products. We mentioned, for instance, the human cell tissue and gene therapy products that come under 351 of the Public Health Service Act. Now, 351 products do not need to follow GMP. They're exempted from this whole thing. However, there is a requirement that people who make any type of 351 product must still register with FDA and must still be on an FDA list of manufacturers. So I would say that basically, to be on the safe side, it's a good idea to register with FDA. Now notice this, registering with FDA does not mean that you are immediately subject to GMPs or subject to inspection by FDA. The registration basically says that, hey, we're in the business of making this product. And now the catch to it is this: if you lose the exemption for some reason, say you're making stuff for a phase 1 product, and then your customer goes into a phase 2 study and uses your product. Now, at that point, you, as a supplier of components, become subject to the GMPs. So at that point, it would be good to be already registered. In fact, some of your customers may ask for instance, the easiest one is to ask you if you have a drug, or what do you call those things? It's a registration as a person that makes a drug, not a final product, but it's mainly registers your product with FDA, and it's done in a way to allow you to keep confidentiality of your manufacturing methods. See, otherwise you have to tell at the phase 2 level, you would have to tell the manufacturer how you make your product, and you may not want this out in public. So you can file one of these things with FDA where you tell FDA how you're making the product, but you may not necessarily tell your customer how you're making the product. And FDA will review these things and keep the whole thing confidential so that your information doesn't get out. Now, issue a GMP, is it appropriate to issue a GMP compliance statement with phase 1 drug products with or without registration with FDA? How does GMP apply to clinical environments where the final drug product is prepared and administered to research subjects? Keep in mind, single batch is prepared for immediate administration. Okay. Yeah, this gets into something very similar to out-and-out cell therapy. Okay, here's the thing. Your statement of GMP compliance, remember, there is no requirement for you to state that you comply with GMP. So if you do this, you are basically declaring to the patient, to the clinical people and all this, that your company is following the GMPs. And this is not, there's not a requirement under the regulations. However, it becomes a contract sort of between you and your customer. Now, this guidance document, the new GMP, in clinical environments where you prepare the drug and administer. Now, remember, research subject. The term research subject is the same as a clinical trial subject under IND. You know, the INDs are considered to be permission to conduct research on humans. That's basically what you're looking at there. So you say administered to research subjects. Well, you're still in clinical trials. And remember this, anytime you administer a drug to a human being, you are into a clinical trial. You know, people, you know, you see these things in the movies about where some guy goes in the lab and whips up something and, you know, he usually transfers it to a syringe without following aseptic procedures or anything, and then goes and stabs a patient with the thing. This is totally uncontrolled. And under food and drug law, you know, you are totally liable and you have violated all sorts of regulations at that point. So the catch to it is this. If you're dealing, you know, the preparation of that final drug product in the hospital or the clinical environment should still follow the guidance document or the so-called companion guidance document that we just talked about. You see, just because, you know, this is the situation of a small laboratory operation where you're preparing phase 1 product. And that's what one of the things that these GMPs were designed to accomplish was that it doesn't, if you look at it, it's not set up for people with large industrial operations. One of the targets here are these small clinical investigations, so-called, where you make product. And, you know, if you're in cell therapy, for instance, it's well known that every prep you make is a new lot. And in some cases, well, for instance, the rules for how many sterility samples you need to take or all this sort of stuff, they're no longer in the GMPs as far as these phase 1 products are concerned. It's not like you were making a larger batch and going into phase 2 studies. Now, with cell therapy studies especially, it's well known that you may need to administer the totality of your product to the patient. I mean, you know, you have this direct relationship between the number of cells you give to the patient and the clinical efficacy. So in these cases, you may not have very much of a retention sample. But if you can't keep a retention sample of the final product, I will suggest to you that you keep retention samples of everything else around, so that if a question comes up about, for instance, the sterility, the purity of the material that you administer to a patient, you'll be able to go back and check these preparations to see where the contaminant may have come from. Now, you know, your single batch prepared for immediate administration, these things are known to FDA. For instance, there are some radiotherapy products that are made. And I know of one situation where the reactor is literally less than a block away from the clinic. And when certain isotopes are made, they are literally transported by runner to the clinic, where they're converted into a form that's suitable for administration and then given to a patient. So these things are not they're not new, and FDA does have requirements for them. So I hope I answered your question without beating around the bush too much. But basically, yes, you do need to follow the new clinical, the 2008 companion guidance.
Thank you, Dr. Guerra. We do have some more questions. A couple of them on your screen right now.
Full development of generic that requires a phase 1 bioequivalence study is the no. Now, here's the catch. A generic, yeah, let me take that back. A generic means that the thing's been licensed and it's been out there. So the expectation is for full GMP compliance. And well, your company, depending upon where you're located, you may not necessarily be registered. You know, this is the thing. FDA registration can happen in two ways. One, you tell FDA that you're manufacturing a certain type of product. See, at this stage, if you're just making a phase 1 product, if it's a completely new product, FDA may not be aware of its existence until you tell them. Or the second way they're going to become aware of its existence is if somebody, your customer, for instance, mentions it in an IND submission. And in this case, with a generic, the product has been licensed. It's out there. So you're not under these new phase 1 GMPs. You're actually under the full GMPs.
Thank you, Dr. Guerra. Just a couple of more questions. Just one moment. Next question should be on your screen now, Doctor.
Okay. Phase 0, similar to, yeah. Okay. Phase 0 for a new product. See, phase 0 in some ways is what, these early phase products that haven't quite reached the phase 1 IND level. And, a lot of these studies, nonetheless, it still goes into a human being. And that's the catch here. See, it doesn't make any difference if they're normals or Superman or, or prisoners for that matter. The fact that it goes into a human being tells you that the product should be GMP compliant. Now, for a new material that has never been commercialized or even gone into phase 2, 3 studies, the appropriate compliance is with these new GMPs, the ones that came out in 2008. Now, the problem here is that it kind of depends upon what you're defining as a phase 0 study. And there's all kinds of studies that, show up under a phase 0. But at this stage, I would imagine that perhaps you haven't yet talked to an FDA investigator about it, or rather a reviewer. And so you haven't gotten any guidance on this sort of thing. And here's one of the big problems that are encountered by many companies, including generics companies, which is that the material that is used in your preclinical studies, in other words, in your animal studies, when you go into humans, in your IND, in your CMC sections especially, you need to be able to show that the material that is used in the GLP studies in animals is essentially the same as the material that you are going to use in your human studies. So this is where, you know, your documentation and controls and everything comes in. And so, yeah, at phase 0, if you're going to go into humans, and, you know, I assume you are, then yes, you need to be GMP compliant, but you don't need to be compliant with 211, but you should be compliant with the companion guidance. Okay.
Thank you so much, Dr. Guara. There's one more question.
Okay.
That's on your screen now.
What level of supplier qualification for API suppliers, for excipient suppliers? Okay. The level of qualification here, and for API suppliers, I assume that you, whoops. Okay, I assume that for API supplier, you're talking about the people that supply material for making the API, right? And then also for excipient suppliers. Now, the excipients go into final product, presumably, or are used in the manufacture of the final product. So the level of qualification there would be the same as for any other component of the final product. Now, for the API suppliers, this can be an interesting situation because many of the API manufacturers themselves do not have good control over the people who supply them. And there's a problem here because your API supplier in turn needs to check his suppliers and do the qualification. Now, the level of supplier qualification really comes down to what would be accepted for that particular product. For instance, if you're getting sodium chloride, for instance, you still have to know what the levels of various contaminants are. And you know, if you can say, well, you know, we go to a reputable company, a well-known company that sells products that are used, you know, like table salt, for instance, and they've been around for years, and they have full testing procedures, and they supply us with a good certificate of analysis and all this. Yes, you know, that can be sufficient. And of course, you still need to do the identity testing of sodium chloride to make sure that what you bought as sodium chloride really is. And let me tell you, in my career, I have had 2 instances where the contents of the bottle were not the same as what was specified on the label. In one case, this is because the bottle had been inappropriately stored and the contents had been degraded. And in another case, it was just an incorrect fill. And in both situations, however, we thought we were dealing with reputable suppliers. And then when we went back to the supplier, they corrected the, you know, the deficiency immediately. But the catch here is to be able to verify that your supplier has a quality system and that the quality system was in force at the time they made your product. Now some companies use a questionnaire, and it's very interesting because some companies will not even respond to a questionnaire, and I think which is pretty poor because, you know, but they say that they don't want to take the time, that it's not worth their while to make the effort to fill out the questionnaire, because sometimes these questionnaires can get, you know, almost ridiculously long. And the other thing that's known about questionnaires is that people lie. They will say, yes, we do this, when in fact they don't do it at all. And so the level of qualification for a critical supply for something that is very important to your product, and whether it's an API or a finished product, I would suggest doing a physical inspection. Just go there and look at the place and see, do they really manufacture in clean facilities? Do they have the SOPs and everything that they claim to have? Now, for excipient suppliers, in some cases, for instance, if you're using lactose and or a one of these corn starches, you can buy it from a company that supplies food grade material. And if you're using the excipient in an oral dosage form, at that point you don't need to qualify the supplier much more than to verify that it does come from the right company and it's made according to certain standards. And let's face it, food grade standards can be just as extensive as pharmaceutical. The problem that will come up is if there's an issue with your excipient. For instance, an excipient that's used in your in a parenteral drug. I once got into a situation where our product started to precipitate. And this was a protein solution, and we were actually adding human serum albumin as a stabilizer to the active ingredient. And when we started to analyze the precipitant, the precipitate rather, we figured out that the precipitate contained mostly denatured human albumin. And the question became what's causing the denaturation? And it was traced back to one of the excipients where we discovered that the excipient that we were using contained an unusually high level of peroxide. And, this took quite a while, actually, a couple of months of study and everything. And then we went on and tried to find a new excipient supplier, only to discover that there was only one manufacturer of this excipient in the U.S. anyway. And he was supplying 3 different suppliers, who in turn supplied it to us. So what happened there was that not only did we have to test lots for peroxide levels, but in one case, we actually had to develop a method of reducing the peroxide because we couldn't purchase material with low peroxide that would, avoid the precipitation problem. So this excipient was completely acceptable to people who were making food products. But in the case of our product, this excipient was not acceptable, and in fact, we had to process it to make it acceptable. So you still have this situation that the excipient supplier, it's sort of a level of how much confidence do you have in the excipient supplier. And if you end up in a situation where the supplier is somebody that you really don't know, and is not really known that well in the industry, and in some cases, trying to conceal themselves behind the intermediate supplier, you really need to start asking questions. And sometimes, for instance, a lot of the recent scandals, especially in China and everything, have dealt with things like diethylene glycol that are used in place of glycerin, where glycerin was an excipient. And, all of these things. So the excipient itself cannot be just considered to be innocent and acceptable just because it's food grade. You really may need to go in and take a close look at it. Anyway, I hope that answers that question. it's one of these answers of it depends. Okay.
Thank you so much, Dr. Kuwara. Well, at this point of time, I would like to thank all our participants for asking these questions. I don't see any more questions coming up. I just want to remind everyone that the polling panel is open. The feedback form has about 8 questions, mostly multiple choices in nature. It wouldn't take 2 minutes of your time to complete them. It would be open till the end of the session. Well, Dr. Kuwara, do you have any parting words before I close today's session?
No, I think that pretty much settles things. Like I said, if you have any questions in the future, please feel free to send them to Global Compliance Panel, and they will forward it to me. Thank you very much for being here.
Thank you so much, Dr. Kuwara. Well, ladies and gentlemen, we are grateful to all of you for having taken part in today's webinar. And if you'd like to get in touch with us, you can send us an email to webinars@globalcompliancepanel.com, or you can also visit our website. Well, we have one webinar that's coming up by Dr. Kuwara early next month. You can log on to our website, which is www.globalcompliancepanel.com for more information, or you can call us at our toll-free number, which is 1-800-447-9407. I would also like to inform all our participants that in case your friends, colleagues, team members might benefit from this webinar, we are happy to inform you that it will be available in a recorded format. We also do have a collection of webinars by Dr. Stephen Kuwara. You can get more information on the same by logging onto our website or calling us at 1-800-447-9407. We welcome your suggestions and feedback or ideas on how we can improve our webinars, or if you would like to suggest a topic or desire a customized corporate training program, online or on-site, we ensure whatever your training necessity is, it would be our priority. We look forward to having you with us again sometime soon, and for your continued patronage. On behalf of the Global Compliance Panel team and Dr. Kuwara, we'd like to thank you for participating in today's webinar, and we wish you a pleasant day ahead. Thank you so much, Dr. Kuwara.